Have you ever paused to truly wonder what makes you, you? Not just your unique personality or, your memories, but the breathtakingly intricate microscopic world humming inside every single one of your cells. Today, we're not just scratching the surface. We're launching a full-scale deep dive into that incredible cellular metropolis. Our mission today is to decode the cell's ultrastructure, the unbelievably complex inner workings of animal cells. We've poured over some fantastic source material packed with mind-boggling details, and our challenge really is to distill it all down. Imagine you're a first-year medical student grappling with this dense subject, and your goal is to understand it so profoundly that you could explain it with clarity and maybe some engaging metaphors to a curious 10-year-old. Think of us as your expert guides, here to help you build those crucial anchors and metaphors, turning complex biology into clear, unforgettable insights.
That's the ultimate test of true understanding, isn't it? Moving beyond just memorizing names and functions to grasping the, elegant choreography of life's fundamental building blocks, seeing how they work together in exquisite harmony. This isn't just academic knowledge, it's the bedrock for understanding everything from why you can think and move to what happens when things go wrong in disease and, how we might intervene with treatments. Simplifying it isn't dumbing it down, it's actually elevating your comprehension to a whole new level.
That's such a powerful way to frame it. For anyone stepping into the medical field, this isn't just about memorizing facts, it's about understanding the foundational machinery of health and disease. So let's pull back the curtain on this microscopic world. From the cell's command center, through its energy hubs, its bustling transport lines, and even its dedicated cleanup crews, ready to truly dig in and discover.
Let's do it.
Okay, let's unpack this journey by starting at a slightly larger scale before we dive right into the cell itself. Our sources begin with a fundamental concept. What exactly is a tissue? It's a term we hear a lot in biology, but what does it really signify?
Right. A tissue isn't just a random assortment of cells sort of haphazardly thrown together. No. It's a very deliberate functional arrangement of specialized cells working in concert, united by a common structure and a very specific task within the larger organism. Think of cells as individual, incredibly versatile Lego bricks. Tissues, then, are like the sophisticated structures you build from those bricks. Maybe a sturdy weight-bearing wall, a flexible joint, or maybe a sensitive control panel. Each built for a particular job, a specific purpose. The scientific study of these tissues, understanding their microscopic structure and how they're organized, that's what we call histology. It's truly the architecture of life, revealing how simple components form complex functional units.
That's a fantastic anchor. So individual cells are our Lego bricks, and tissues are the actual purposeful structures built from them. Maybe a robust wall-like skin, or a transparent window pane in your eye, or flexible support beams and joints, each with a distinct critical function. And our sources tell us there are 4 fundamental tissue types, 4 main categories that form everything in our bodies. But what makes these 4 so fundamental? Is it like their embryonic origins, or just they're everywhere?
Well, it's absolutely a combination of both, really. These 4 distinct types actually emerge from very specific embryonic cell layers during development, and together they comprise the entire functional architecture of our bodies. They are the universal building blocks, if you will, present in varying proportions and arrangements in every organ and system. Let's quickly outline them. First, epithelial tissue. That's your protective covering. It forms linings and surfaces, like the skin shielding you, or the delicate lining of your internal organs, controlling what passes through. Next, we have muscle tissue, the engine of movement, responsible for everything from lifting something heavy to your heartbeat to the subtle movements of your gut. Then there's nervous tissue, the communication network. Imagine the intricate, high-speed wiring of a supercomputer that's nervous tissue, rapidly sending signals, coordinating every thought, sensation, action. And finally, connective tissue. Or support tissue. This is the glue, the framework, the filler. It holds everything together, provides structural support like steel beams, stores energy, protects organs. Think bones, cartilage, blood, the fibrous tissue binding muscles to bones. And they're not just separate, they're in constant, intricate collaboration, each absolutely essential for an organ's complete function. Now, diving deeper, one of the most profound organizational principles our sources highlight is how membranes create compartmentalization within cells. Why is this internal division so incredibly crucial for eukaryotic cells, especially compared to, say, simpler cells?
It's like having separate rooms in a house, right? You cook in the kitchen, sleep in the bedroom. You don't want those activities mixing.
Exactly. That analogy is spot on. Eukaryotic cells, the cells making up us and most complex life, are vastly more complex than simpler prokaryotic cells like bacteria, which generally lack these internal membrane-bound divisions. This segregation, this compartmentalization by membranes allows literally thousands of distinct biochemical reactions to happen in an orderly, controlled fashion simultaneously within the same tiny cell. Imagine trying to run a bustling factory floor without any separate workstations, no walls, all the raw materials and finished products mixed together.
Yeah.
Chaos. Absolute mess, leading to inefficiency, cross-contamination. Nothing would get done properly. By having these distinct compartments, the cell can, first, optimize conditions like pH or enzyme concentrations for different reactions. Second, concentrate specific enzymes or substrates exactly where they're needed. And third, prevent destructive enzymes from, wreaking havoc where they shouldn't. It essentially allows the cell to run a highly organized and incredibly efficient internal economy.
So compartmentalization is basically like having different specialized workshops or secure rooms in that factory. Each room with a specific job, ideal conditions. Keeping things separate prevents interference, improves efficiency, and allows for much more complex operations. A genius design.
Absolutely.
Now, our sources emphasize that most cellular components, the organelles, these tiny internal organs, can only be seen in detail with an electron microscope. Why can't we just use a regular light microscope? We can see bacteria with those, right? So why not the tiny bits inside our own cells?
That's a great question. It really gets to the heart of how we visualize this microscopic world. It all comes down to something called the limit of resolution. This is the minimum distance at which you can distinguish two separate objects as, distinct entities. If they're closer than this limit, they just blur together no matter how much you magnify. A traditional light microscope using visible light waves has a theoretical limit of about 0.2 micrometers. That's perfectly fine for seeing whole bacteria, which are maybe 110 micrometers. And you can even make out larger organelles like mitochondria around 0.5 micrometers. But many, many organelles, and certainly the fine, intricate details within them, are much, much smaller, often down to nanometer scales, thousands of times smaller than micrometers.
So to really peer into that hidden world, we need something vastly more powerful. That's where electron microscopy, EM, comes in, like a superpowered magnifying glass, seeing in a whole new way.
Precisely. Electron microscopy revolutionized biology. It uses a beam of electrons instead of visible light. The key here is that electrons have a much, much smaller wavelength than light. That's the secret to their superior resolution. The shorter the wavelength they use, the smaller the limit of resolution, and the more minute detail you can resolve. For biological samples, we mainly use two types. Transmission electron microscopy, TEM, resolves details down to maybe 2 nanometers. This lets us see through ultra-thin sections of cells, observe internal ultrastructure organelles, membranes, fine details with incredible clarity. Then there's scanning electron microscopy, SCM. This gives a stunning high-resolution surface view, a 3D perspective, with resolution around 10 nanometers. So imagine trying to read tiny print. A regular magnifying glass, the light microscope, helps a bit. But for the truly microscopic details, the ultrafine print of the cellular world, you need that superpowered magnifying glass using electrons. That's your EM, revealing a hidden universe.
Fascinating. And forming all these compartments, these internal walls and the outer boundary, are membranes. Our sources talk about unit membranes and the fluid mosaic model. What exactly is going on with these membranes?
Right. All cell membranes, the outer plasma membrane or internal organelle membranes, share a basic structure. They're fundamentally built from a phospholipid bilayer. These phospholipid molecules are quite special. They're empapathic, a great word meaning they have both a water-loving hydrophilic head and two water-fearing hydrophobic tails. Because of this dual nature, in a watery environment, like inside or outside a cell, they spontaneously arrange into a double layer. The water-fearing tails point inwards, shielded from water, forming the membrane's core. The water-loving heads face outwards, interacting with the water on both sides. Under TEM, these bilayers look like 3 distinct layers, 2 dark lines, the heads, separated by a light layer, the tails. This trilaminar look led to the unit membrane concept.
And this isn't just a static wall, is it? The fluid mosaic model sounds incredibly dynamic.
It truly is. The fluid mosaic model proposed by Singer and Nicholson beautifully captures this. It suggests these phospholipid molecules aren't rigidly fixed, but are constantly moving, exchanging places, rotating, flexing, giving the membrane a fluid, almost oily consistency, like olive oil. Within this fluid bilayer, many different proteins are embedded or associated, like diverse, colorful tiles in a mosaic. These proteins float freely in the lipid sea. Some integral proteins are embedded or span the bilayer, forming channels or receptors. Others, peripheral proteins, are associated with one surface. And many are glycosylated. They have short, branched sugar chains attached, especially on the outer surface. Adding another layer of complexity. These sugars and proteins can also interact with the cell's internal skeleton, the cytoskeleton, providing anchorage and shape.
And the outermost layer, the cell's skin, separating it from the outside world, that's the plasma membrane.
Indeed, the plasma membrane, or plasmalemma, the outermost boundary, typically around 9 nanometers thick. On its outer surface, those sugar chains we mentioned from glycoproteins and glycolipids form a fuzzy protective coat called the glycocalyx. This glycocalyx is incredibly important for cell-to-cell recognition, adhesion, protection. It's like a unique ID badge or fingerprint, allowing cells to identify each other, determine self from non-self, interact appropriately to form tissues. It also protects the cell surface from mechanical and chemical damage.
So far more than just a barrier. It's a sophisticated communication hub, a selective gatekeeper, an identity card all rolled into one.
Absolutely. Its functions are incredibly diverse and vital. It's involved in intercellular adhesion, helping cells stick together, crucial for recognition, like in immune responses or development. Facilitates signal transduction, receiving messages from outside, hormones, growth factors, and relaying them inside. Of course, maintains compartmentalization, keeping inside distinct from outside. It's selectively permeable, choosing precisely what goes in and out, controlling passage of ions, nutrients, waste, involved in various transport types, active and passive, and mediates processes like endocytosis, bringing things in by engulfing them, think a white blood cell eating bacteria, and exocytosis, expelling substances out like secreting hormones. Essentially, the cell membrane is like the skin of a water balloon, but way, way smarter, dynamic, made of tiny oily bubbles, phospholipids loving to stick together. Floating in the smart skin are little doors, channels, sensors the proteins, deciding who gets in, who gets out, who the cell talks to. And that glycocalyx, its unique ID badge or fingerprint, constantly signaling.
Okay, moving inwards. Here's where it gets really interesting. The central processing unit, the nucleus. Our sources confirm it's almost always the most prominent cellular structure. It definitely stands out.
Absolutely. Prominent and indispensable. It's the main library, the control room, the master architect's office, all rolled into one secure location. It contains the cell's entire genomic DNA, all the incredibly detailed blueprints and instructions for making the cell, directing its activities, ensuring proper function and replication. This is also where mRNA transcription primarily happens, meaning specific copies of those genetic blueprints are made into messenger RNA, which then carries instructions out to the protein factories in the cytoplasm. It's the ultimate command center, orchestrating the entire life of the cell.
And when we look at this genetic material under EM, we see different patterns, right? Suggesting different states of activity.
Yes, that's a key observation. TEM reveals the DNA tightly wound around proteins called histones, forming chromatin. We see two main forms. First, electron-dense heterochromatin, dark and clumped. This DNA is tightly packed, generally inactive. Genes silenced, not being transcribed, stored away. Second, electron-lucent euchromatin, lighter, more dispersed. This DNA is less condensed, accessible for transcription, actively involved in RNA synthesis. It's a great visual indicator of a cell's metabolic activity. An inactive cell, smaller nucleus, more heterochromatin. A very active cell, like a neuron or gland cell. Larger nucleus, dominated by euchromatin, indicating intense RNA and protein production. So think of the nucleus as a huge library. Heterochromatin is the vast collection, neatly shelved, not currently read, stored but available. Euchromatin is like the books open on desks, actively used, copied, ready for information to be extracted.
And this library isn't just open to the elements. It's protected by a double-membrane nuclear envelope. Sounds like serious security.
It absolutely is. This nuclear envelope is actually a specialized form of the endoplasmic reticulum, which we'll get to. This double membrane has a space in between, the paranuclear cisterna, which is continuous with the ER's internal space. So it's connected, like two layers of security walls around the library, protecting the genetic contents from the cytoplasm. But these walls aren't sealed. They contain crucial nuclear pores. Not just holes, but highly organized protein complexes, octagonal. These pores act as incredibly selective gates, controlling precisely what goes in and out, regulating export of ribosomal subunits, mRNA, specific proteins, and importing necessary proteins like histones. The whole envelope is supported internally by a dense network of intermediate filaments called the nuclear lamina, helps maintain shape, provides strength, plays a role in chromatin organization. So yes, a double-layered security wall with sophisticated guarded gates, the nuclear pores, meticulously controlling information flow.
And inside the nucleus, nestled within, there's another important structure, the nucleolus, like a smaller workshop within the library.
Indeed. The nucleolus is fascinating, a non-membrane bound structure serving as the cell's dedicated ribosome assembly plant. This is where ribosomal RNA, rRNA, is synthesized, processed, and where initial ribosome assembly occurs. Combining rRNA with ribosomal proteins imported from the cytoplasm, it's incredibly busy. Cells active in protein synthesis neurons, gland cells, tend to have very prominent, large nucleoli, indicating high demand for ribosomes. Once assembled, these ribosomal subunits are exported through the nuclear pores to the cytoplasm, ready to build proteins. So inside the library, the nucleolus is like a dedicated, bustling workshop, manufacturing and partially assembling all the tiny robots' ribosomes before sending them out to the factory floor.
Our sources actually drop a surprising fact. Not all cells in the human body keep their nuclei. Seems fundamental, but apparently not always. What proportion are nucleated, and which ones break this rule, and why?
It's a great question, highlighting incredible cellular adaptation and specialization. The vast majority of your cells are nucleated, carrying that essential blueprint. However, there are key exceptions, where the nucleus is ejected during maturation or just never forms. Adapted for specific functions where it would be a hindrance. The most prominent example, mature red blood cells, erythrocytes. They eject their nucleus and other organelles during development. To make more space for hemoglobin allows them to carry maximal oxygen, but means limited lifespan, no repair, no new proteins. You also find no nuclei in the outermost skin layers, the stratum corneum cells, essentially dead keratin-filled protective flakes, a robust barrier. And another fascinating example, lens fiber cells in the eye. Absence of a nucleus allows greater transparency, crucial for clear vision. In these cases, the cell optimized its design by removing the nucleus. Evolution finding peak performance for a specific role.
Incredible adaptation. Okay, back to our typical cell. That nucleus, with its double wall, the nuclear envelope, it's physically connected to our next stop, the endoplasmic reticulum, ER. Literally inside plasma network. Sounds like a sprawling internal highway system. What's this massive network all about?
You've hit on a perfect description. Think of the ER as a vast interconnected network. Membrane-bound tunnels, flattened sacs called cisternae, and small vesicles. This whole network is continuous, stretching throughout the cytoplasm from the nuclear envelope, forming one single large internal compartment, the ER lumen, or cisterna. It's the cell's major internal transportation and processing hub, deeply integrated into almost everything. A quality control center, synthesis factory, storage depot, all at once. And critically, it comes in two distinct flavors, different structures, specialized jobs.
Two types. Okay, first the rough ER. What makes it rough, and what's its job in our cellular factory?
Yes, the rough is fundamental to its function. Under EM, the rough endoplasmic reticulum, RER, appears started with thousands of tiny particles on its outer surface. These are ribosomes, giving it that characteristic rough, bumpy look. The RER is incredibly abundant in cells actively synthesizing lots of proteins, especially those for secretion out of the cell for integration into membranes or for delivery to lysosomes, pancreatic acinar cells, churning out digestive enzymes for secretion, packed with RER, plasma cells making antibodies, also extensive RER. The sheer abundance of membranes and ribosomes makes these cells stain strongly with basic dyes like hematoxylin, visible even under a light microscope.
So its main job is protein synthesis and initial modification, like a factory floor with tiny robot workers' ribosomes attached, building specific products, proteins, and immediately moving them inside the factory's tunnels for processing or shipping. What kind of processing happens in there?
Exactly. Once a protein starts synthesis on a ribosome that docks with the RER, it's threaded into the ER lumen. Inside, a lot happens. Proteins undergo initial folding, often helped by chaperone proteins ensuring correct 3D structure. They might get sugar chains added, glycosylation helping with targeting and function. Disulfide bonds form, critical for stabilizing structure. This quality control is stringent. If proteins don't fold right, the RER has a system called the unfolded protein response, UPR. UPR tries to correct misfolding. If it fails, it triggers pathways for protein degradation or even programmed cell death. Malfunctions in UPR are actually linked to several diseases like Alzheimer's and Parkinson's, where misfolded proteins accumulate.
Wow, so not just making proteins, but ensuring they're perfect and dealing with rejects, advanced factory. And then there's the smooth ER, without ribosomes, different function. Or complementary.
A completely different yet complementary set of functions. The smooth endoplasmic reticulum, SER, typically less expensive than RER in most cells. Its cisternae aren't as flattened, more tubular, interconnected. Less protein factory, more versatile chemical lab. Crucial for diverse metabolic processes. SR is critically involved in lipid biosynthesis, making fats, including phospholipids for new membranes, and cholesterol, a precursor for steroid hormones. Vital for steroid production in endocrine cells, ovary, testes, adrenal gland, synthesizing hormones like estrogen, testosterone, cortisol, a very important role, storing and regulating calcium ions, especially abundant in muscle cells where it's called sarcoplasmic reticulum. Rapid release and reuptake of calcium from SR triggers muscle contraction and relaxation.
So making fats, hormones, and acting as a crucial calcium reservoir.
Precisely. And perhaps most famously, especially in the liver and kidneys, SER is heavily involved in detoxification. Contains enzymes called cytochrome P450. They metabolize and detoxify various harmful molecules, alcohol, pesticides, many drugs, breaking them down into less toxic, more easily excretable forms. Our sources highlight about 50% of ethanol consumed is detoxified this way in the liver's SCR. So SCR is like a separate specialized chemical lab. Lipids crafted, steroids processed, calcium stored and released, harmful chemicals neutralized, keeping the cell and organism healthy. No noisy robots here, just quiet, efficient, vital chemical reactions.
Let's zoom in on those tiny protein builders, the ribosomes. Found on the RER and floating free, what are these little machines made of, and how do they build proteins from a genetic message?
Ribosomes are truly fascinating molecular machines, essential for every cell. They are electron-dense particles, quite small, 20-30 nanometers, not enclosed by a membrane, composed of ribosomal RNA, rRNA, and about 75 distinct proteins. Two main parts. A large 60S subunit and a small 40S subunit. The S is Svedberg unit, a measure of sedimentation rate, basically size and density. These two subunits only come together when they attach to messenger RNA, mRNA, the blueprint copies from the nucleus. To start the incredible process of catalyzing protein synthesis, or translation, they literally translate the genetic code from mRNA into a sequence of amino acids, which then folds into a functional protein.
So these tiny builders are universal, but what determines where the protein ends up? Inside the cell or shipped out? Is it just where the ribosome is, or some other signal?
That's critical, and it's primarily about where the ribosome is when it starts translation, which is itself controlled by signals in the mRNA and the protein being made. We have two main populations leading to two main destinations. First, free polyribosomes, or polysomes. Clusters of ribosomes floating freely in the cytoplasm. These synthesize proteins destined to remain inside the cell. Structural proteins for the cytoplasm, metabolic enzymes, proteins for the nucleus, peroxisomes, mitochondria, building parts for the cell's own internal use. Then we have the membrane-bound ribosomes, the ones attached to the RER. The key here is a signal peptide, a short amino acid sequence at the protein's beginning. As this emerges, it's recognized by a signal recognition particle, SRP, which pauses translation and escorts the ribosome to the RER membrane where it docks. These ribosomes synthesize proteins destined for cell membranes, for lysosomes, or crucially, proteins to be secreted outside the cell.
Okay, so ribosomes are like the cell's 3D printers, working from mRNA blueprints. If free-floating, they print parts for internal use, staying right there. If attached to the RER, the factory wall, they print products installed into the wall, sent to recycling, or shipped out entirely. An incredibly precise production and delivery system.
Exactly.
Once proteins are made, especially on RER, they often head to another major organelle.
Okay.
The Golgi apparatus. What's its role? Sounds like the cell's post office, customization shop, sorting center, all rolled into one.
That's an excellent multi-part analogy. The Golgi absolutely is all those things.
Right.
A central hub for modifying, sorting, packaging proteins and lipids for secretion or delivery to other organelles. It consists of stacks of flattened, membrane-bound sacs called cisternae, often described as saucer-shaped, like stacked pancakes not quite connected. Proteins and lipids from the RER arrive in small ER-derived transport vesicles. These bud off the ER, migrate to the Golgi, fuse with its receiving face, delivering cargo for processing.
And it's not just one stack. It has functional zones, right? Like different stages of manufacturing and shipping.
Yeah.
Directionality.
Precisely. The Golgi shows distinct polarity. Functional regions representing a clear flow. There's the nuclear-facing cis face, or cis-Golgi network, the receiving end, where ER vesicles arrive, like the receiving dock. Next, cargo moves into the medial Golgi, the central processing unit. Many critical modifications happen here, further glycosylation, adding different sugar chains, other chemical mods like phosphorylation, the customization shop. Finally, the trans-Golgi network, TGN, the shipping department. As proteins migrate through, they undergo sequential modifications, are carefully sorted based on final destination, then packaged into new vesicles budding off the TGN. This precise sorting ensures each protein reaches its correct cellular address, The Golgi also synthesizes components of the extracellular matrix, like proteoglycans, secreted outside to help build tissues.
So the Golgi is like a series of assembly lines. Raw products from the ER come to the cis-face receiving dock, move through medial Golgi assembly, customization, maybe getting tags, then exit from trans-Golgi network shipping department, carefully packaged. And our sources mention Golgi vesicles tagged with coat proteins, like special stamps. These vesicles are the delivery vehicles, tiny trucks.
Exactly, you've nailed their role. Vesicles are tiny membrane-bound sacs budding off organelles, the cell's internal transport system, like little delivery trucks or drones, each tagged with specific coat proteins on their surface. These coats like clathrin, CoPI, CoPII, for different steps act as address labels, ensuring the package, the cargo, gets delivered precisely where needed. This specificity is absolutely crucial for maintaining cellular organization.
And our sources list several types of these crucial delivery vehicles.
Yes, several main types, each specialized. First, cell surface-derived endocytotic vesicles, bringing things in from outside. Penocytotic vesicles for fluids, cell drinking. Phagocytotic vesicles, larger engulfing particles like bacteria. Think macrophages. Then, Golgi-derived transport and secretory vesicles, carrying processed protein lipids to their destination. Plasma membrane, lysosomes, or secretion outside the cell, hormones, enzymes. Also, ER-derived transport vesicles, carrying newly synthesized protein lipids from ER to the Golgi. And finally, two very important specialized types, lysosomes with digestive enzymes for recycling and peroxisomes with oxidative enzymes for detox. So vesicles are indeed like small specialized delivery drones or trucks, picking up packages, having specific labels, coat proteins, delivering precisely, ensuring internal logistics run with astonishing smoothness.
Now this next one is incredibly important, produced by the Golgi, playing a huge, maybe unglamorous but vital role. The lysosome, what's its big job? Why is it so critical?
Lysosomes are aptly called the cell's recycling centers, waste disposal units, even its gastric system. They are membrane-bound organelles packed with a formidable arsenal of powerful digestive enzymes, acid hydrolases, about 40 different types. Nucleases for nucleic acids, proteases for proteins, lipases for lipids, phosphatases for phosphates, designed to break down a vast array of cellular waste, worn-out organelles, materials brought in from outside. Crucially, these enzymes work best in a very acidic environment, pH around 4.5 to 5.0, which the lysosome actively maintains by pumping protons, H+A, across its membrane using a specialized pump.
So super acidic, highly destructive inside. How does the lysosome itself avoid being digested? Seems risky.
Excellent point. Testament to elegant design. The lysosomal membrane is adapted for protection. Its inner surface, facing the acid, is heavily glycosylated. Membrane proteins have lots of sugar chains attached, forms a thick, protective glycocalyx on the internal surface, a sugary shield, preventing the membrane being digested by its own enzymes. Essential safeguard. We also distinguish different states. Primary lysosomes are newly generated by Golgi, dense, spherical, dormant, ready. Secondary lysosomes, or phagolysosomes, sendolysosomes, are the active digestive units, formed when primary lysosomes fuse with other vesicles like phagosomes, containing outside material, or autophagosomes, containing worn-out internal parts. It's within these secondary lysosomes that actual digestion happens.
So lysosomes were like self-contained recycling bins or shredders, powerful acidic digestive uses breaking down waste. And their inner lining is extra tough, coated in sugar to protect themselves. Internal defense.
Exactly.
Our sources describe a fascinating vital process related to the lysosomes called autophagy, literally self-eating. Sounds intense.
It does sound dramatic, but it's actually a fundamental, finely tuned, vital, homeostatic process for health, renewal, survival. Autophagy is the lysosomal breakdown of non-functioning organelles, damaged components, even invading pathogens. It's how the cell cleans house, preventing toxic junk buildup, and crucially, recycling digested products, amino acids, fatty acids, nucleotides back into useful building blocks or for energy. Ensures efficiency, quality control, allows adaptation to stress or starvation. The process involves a membrane, often from the SER, wrapping around the old damaged part, forming a double membrane autophagosome. This autophagosome then fuses with a lysosome, contents broken down, recycled, incredibly important. Elucidated by Professor Yoshinori Osumi Nobel Prize in 2016 for this work, highlighting its profound significance.
And significant clinical relevance too, I imagine, given its role in cleanup.
Absolutely vast and growing. For instance, under stress like starvation, cells ramp up autophagy to self-digest parts for energy building blocks, usually beneficial. But tumor cells can sometimes hijack this to survive challenging conditions, becoming more resistant to therapy. So inhibiting autophagy is explored as an anti-cancer strategy. Conversely, impaired autophagy is implicated in neurodegenerative diseases. Accumulation of damaged proteins organelles leads to dysfunction. What's left after digestion? Indigestible remnants called residual bodies. If not excreted, they accumulate over time as dense yellowish-brown leukofusin granules, often seen as a marker of aging cells, especially long-lived ones like neurons, cardiac myocytes, can interfere with function.
So autophagy is like the cell's internal spring cleaning or car mechanic. Old part isn't working. Surround it, take it to the recycling center, lysosome, break it down, use the parts again. Masterclass in cellular economy and self-renewal.
Well put.
This brings us to a crucial point for medicine. What happens if these digestive systems, these lysosomal enzymes, don't work right?
This is where we see lysosomal storage disorders, LSDs, devastating conditions. If there are genetic defects, usually inherited in one or more specific hydrolases, their target substrates can't be digested. Leads to pathological accumulation of undigested material residual bodies piling up, often to toxic levels. Causes severe dysfunction, organelle swelling, pathology in tissues, especially the nervous system. While individually rare, collectively they affect about 1 in 7,700 live births, over 70 known disorders, each caused by a specific deficient enzyme.
Can you give a couple of examples to illustrate the impact?
Certainly. Hurler syndrome. Accumulation of complex sugar molecules, glycosaminoglycans, dermatan heparin sulfate, due to a deficient enzyme, leads to skeletal abnormalities, organ enlargement, developmental delay. Another, perhaps more widely known, Tay-Sachs disease. Devastating neurological disorder, lack of lysosomal enzyme hexaminidase A, a specific lipid GM2 ganglioside crucial for nerve membranes, builds up toxically, particularly in brain spinal cord neurons. Disrupts nerve function, leads to progressive neurological deterioration, blindness, intellectual disability, usually death in early childhood. For some LSDs, we have enzyme replacement therapy, delivering the missing enzyme. But many still lack effective treatments. So imagine the recycling truck's engine is broken, or the shredder can't chop plastic. Waste piles up, blocks the factory, causes toxic problems. That's LSDs. The system fails, the internal environment gets overwhelmed.
Another membrane-bound organelle, somewhat similar in breaking things down, is the peroxisome. What's their deal? Another recycling center, or more specialized?
Peroxisomes are fascinating. Best described as the cell's specialized detox squad or chemical neutralization specialists. Small spherical organelles, 0.51 micrometer, bound by a single membrane like lysosomes, found in virtually all eukaryotic cells, particularly abundant in liver and kidney cells, key organs for detoxifying the bloodstream. Their primary role involves oxidative reactions that generate and then immediately neutralize a powerful, potentially damaging byproduct.
So they use oxygen to break things down, but have to be careful with the byproducts. That's right. Major sites of oxygen use. Specialized enzymes, oxidases, take a substrate, RH2, use oxygen to break it down, producing a potent reactive waste product. Hydrogen peroxide, H2O2. Think efficient but slightly messy reaction. RH2 plus O2, Lr plus H2O2. H2O2 is the energetic but dangerous byproduct. Hydrogen peroxide itself is highly reactive, can damage components if it accumulates, so peroxisomes also contain a crucial enzyme, catalase. Catalase is two vital things. Rapidly dismutes H2O2 into harmless water and oxygen, 2H2O2, A2H2O plus O2, and uses the H2O2 they generate to oxidize other toxic substrates, neutralizing them, like phenols, formic acid, formaldehyde, and, critically, alcohol. Sources highlight about 50% of consumed ethanol detoxify this way in peroxisomes, mainly liver. Also particularly important for oxidation of very long-chain fatty acids, C18+, breaking them down into shorter chains for energy processing in mitochondria.
And if peroxisomes aren't working correctly, especially with those long-chain fatty acids,
Just like lysosomes, defects cause severe pathologies. A notable example: adrenoleukodystrophy, ALD. Serious genetic disorder caused by impaired oxidation of those very long-chain fatty acids, VLCFAs, in peroxisomes. Leads to harmful accumulation of VLCFAs, primarily affecting myelin sheath of nerve cells, brain spinal cord, and adrenal glands. Causes progressive neurological damage, learning disabilities, behavioral changes, loss of motor control vision. So peroxisomes are like the cell's hazmat team, taking in toxins, breaking them down, producing a dangerous byproduct, H2O2, but smartly designed to quickly neutralize it with catalase, preventing damage, a continuous, vigilant cleanup.
Okay, now for perhaps the most famous organelle,
Yeah.
the mitochondria. Everyone knows the powerhouses of the cell, but what does that actually mean? What power are they generating and how?
It means they are the primary site of ATP synthesis. ATP, adenosine triphosphate, the universal energy currency of the cell. Without ATP, literally nothing functions. No protein synthesis, movement, active transport, nerve impulses, nothing. Mitochondria are the sophisticated power plants generating the vast majority of this ATP through cellular respiration. They contain essential enzymes for the citric acid cycle, aka TCA or Krebs cycle, and the intricate machinery for oxidative phosphorylation and the electron transport system. These complex processes break down glucose and fatty acids, the cell's fuel, generating vast amounts of ATP. They literally power almost every activity your cells perform.
So a cell with high energy needs, like muscle or liver, must have loads of mitochondria.
Precisely. Mitochondria number is directly proportional to metabolic activity and energy demand. High-energy cells, liver, skeletal muscle, neurons are packed with them. Sources note a single liver cell can have up to 2,000 mitochondria. Shows the incredible energy demands and mitochondria's central role. So mitochondria are like tiny, highly efficient power plants, taking in fuel, glucose, fatty acids, and through complex chemistry, generating all the electricity ATP the cell needs.
And their internal structure is unique, that double membrane. How does that help power generation?
Their double membrane structure is absolutely critical. An outer membrane, relatively smooth, permeable to small molecules, like a perimeter fence. But the inner membrane is remarkable, highly folded into cristae, projecting inwards into the inner matrix.
Right.
These folds dramatically increase surface area for the hundreds of enzymes and complexes in ATP production, especially electron transport chain and ATP synthase. Maximize surface area, maximum efficiency. The space between membranes is the inner membrane space. Innermost compartment is the matrix containing citric acid cycle enzymes.
Here's where it gets really interesting, almost mind-bending. Mitochondria have their own circular genome and translational machinery. What's up with that? Sounds like they were once independent.
This is truly one of the most remarkable features, key to their evolutionary origin. Each mitochondrion contains a small circular genome, like a bacterium, about 16.5 kilobases, encoding 37 genes, specifying some ribosomal RNAs, transfer RNAs, a few critical electron transport chain proteins. They also have their own ribosomes, similar to bacterial ones, 7DS versus eukaryotic ADS, own tRNAs. Can divide autonomously by binary fission, separate from cell division. This suite of bacteria-like traits is incredibly strong evidence for the endosymbiotic theory. Proposes mitochondria originated from ancient free-living alpha proteobacteria. Billions of years ago, these bacteria developed a symbiotic relationship with primordial eukaryotes. Engulfed but not digested, became integral, indispensable, not just a footnote, a revolutionary insight. Reshaped our understanding of complex life's origins. Imagine the astonishment. These powerhouses were once independent organisms, testament to life's ability to form partnerships, deeply intertwined history of bacteria and eukaryotes.
Absolutely incredible. A true aha moment in biology. And a fascinating detail from that origin. Maternal inheritance.
Yes, another striking consequence. You inherit all your mitochondria solely from your mother. During fertilization, sperm contributes virtually no mitochondria. Only the egg provides them. Makes mitochondrial DNA incredibly useful for tracing maternal lineages in genetics, population studies, anthropology. Passed down virtually unchanged through the maternal line. Beyond ATP synthesis, mitochondria have other roles. Key regulators of apoptosis, programmed cell death, releasing factors like cytochrome C when cell is damaged beyond repair, also involved in calcium signaling and steroidogenesis in certain cells, and like other organelles when their genetics or function go wrong, severe disease. Kern-Serre syndrome, for example, mitochondrial disorder from large mtDNA dilutions, causes muscle weakness, heart problems, neurological issues. So mitochondria, mini power plants, once separate organisms, tiny ancient bacteria, joined forces with our cells, still run their own generators with own instructions, DNA powering the whole cell, with internal folded walls, cristae packing in more energy machinery, tirelessly fueling everything.
Okay, so we've got organelles, power, manufacturing.
Yeah.
But how do they keep shape, move around? How do vesicles get from ER to Golgi? Proteins get down long nerve cells. That brings us to the cytoskeleton. Sounds like the cell's internal framework, but I bet it's more dynamic than static bones.
You're absolutely right to emphasize dynamic. The cytoskeleton is the cell's internal skeleton, but far, far more dynamic and adaptable than static bones. A complex, intricate network of protein filaments, constantly assembling, disassembling, reorganizing. This dynamic network is responsible for a phenomenal array of functions, maintaining and changing cell shape, providing robust structural support for plasma membrane and organelles, enabling precise movement of organelles and vesicles, providing locomotor mechanisms for entire cells to move, immune responses, wound healing, contributing to contractility, muscle, truly multi-talented living scaffolding, constantly built and rebuilt.
And our sources say it has three main components, each specialized.
Yes, three main components collaborate: microfilaments, intermediate filaments, and microtubules. Each distinct structures and functions, but all work together, often interacting. So metaphorically, the cytoskeleton is like the cell's internal system of roads, train tracks, scaffolding, provides support, allows movement, helps change shape, even lets the cell crawl or swim, adapting constantly.
First up, the smallest ropes. Microfilaments, mainly actin. Sound like the quick response team.
Great way to think of them. Smallest components, 5-7 nanometers diameter, polymers of globular actin, G-actin assembling into filamentous actin, F-actin, often twisted like a double helix, primarily cortical, concentrated just under plasma membrane, forming the cell cortex, provides mechanical strength, helps define shape.
And incredibly dynamic, constantly building, unbuilding, reshaping on the fly.
Yes. Defining characteristic functional strength associated with ATP, powering assembly-disassembly. New monomers added at plus end, removed from base end, treadmilling. Dynamic nature allows rapid, efficient shape changes, crucial for maintaining shape of intestinal microvilli, increasing surface area. Form temporary protrusions like lamellipodia, filopodia, enabling cell migration, amoeboid movements, cell crawling. Central to contractility, interacting with myosin motors in muscle. Form contractile ring pinching cell in two during cytokinesis, so microfilaments. Thin, flexible ropes. Cell can quickly build, shorten, dismantle. Excellent for quick shape changes, pushing out fingers, helping crawl or pinch apart. Versatile, dynamic engineers of shape and movement.
Next, intermediate filaments. Sound more like robust structural beams, more permanent support.
Perfect analogy. Rope-like structures, 10-12 nanometers diameter, intermediate size. Critically, unlike the others, intermediate filaments are remarkably stable, not dynamic. Provide incredibly robust, long-lasting structural support, giving mechanical strength, resistance to stress, maintaining tissue integrity. What's fascinating and clinically relevant, they are heterogeneous and cell type specific. Specific proteins vary significantly by cell type, like molecular fingerprints.
So different cells use different kinds of intermediate filaments for structure.
Precisely. Useful in pathology for identifying cell origins, example diagnosing cancers. Epithelial cells contain keratin, immense tensile strength skin. Mesenchymal cells, fibroblasts, contain vimentin. Muscle cells contain desmin, integrating contractile units. Neurons have neurofilaments supporting long axons. Even the nucleus has lamins forming nuclear lamina. Main function, form tough, resilient supporting meshwork, anchored to plasma membrane at strong intercellular junctions. Desmosomes, cell-cell, and hemidesmosomes, cell matrix. Riveting cell tissues together.
And mutations in keratin can cause skin blistering, highlighting their vital structural role.
Precisely. Directly illustrates critical role and consequences when integrity is compromised. Mutations in basal keratin genes, K5, K15, cause conditions like epidermolysis bullosa, severe skin blistering, because mutations cause loss of cohesion between basal epithelial cells, can't hold together under minor stress friction, separate, causing blisters. So intermediate filaments, like steel beams or rebar, main strong permanent structural support, specialized internal frameworks unique to cell types, giving tissues strength and resilience.
Finally, the thickest, microtubules, often associated with movement, internal and cellular, the cell's major transport network.
Microtubules are truly the cell's superhighways and train tracks, relatively large, hollow, cylindrical protofilaments, 25 nanometers diameter, assembled from 13 alpha-beta tubulin dimer subunits in a spiral. Like microfilaments exhibit polarity, plus end, beta tubulin exposed and alpha tubulin exposed. Dynamics going faster from plus end, powered by GTP hydrolysis, rapid assembly, disassembly, dynamic instability, growth rescue versus shrinkage catastrophe, typically originate from the centrosome primary microtubule organizing center, MTOC, nucleating and anchoring them.
And these long, rigid tracks are used for cargo transport.
Yes, a crucial function. Precise, polarized tracks for organelle vesicle movement. Specialized motor proteins, dynin and kinasin attached to cargo, organelles, vesicles, chromosomes, literally walk them along microtubule tracks using ATP. Kinins generally move towards plus end, anterograde away from centrosome, dyninins generally towards and retrograde towards centrosome. This is how neurotransmitters made in neuron cell body shuttle efficiently down the long axon to terminals, essential for nerve function, also fundamental during cell division. Form mitotic spindle apparatuses meticulously separating chromosomes into daughter cells, ensuring each gets complete genetic set.
And they form the core of cilia and flagella, those hair-like structures.
Indeed. Form the core structural element axonomy of cilia and flagella, hair-like projections for movement. Motile cilia sweep mucus from lungs. Flagella propel cells like sperm. Classic 9+2 arrangement of microtubules, 9 doublets around 2 singles. Rhythmic beating powered by dining arms causing microtubules to slide. Also primary cilia, non-modal, usually 9+0, act as sensory antenna, sensing chemical and mechanical signals. Malfunctions linked to polycystic kidney disease, Tay-Sachs inversus. So critical that drugs targeting them, colchicine, gout, Taxol chemo, work by interfering with assembly-disassembly, highlighting vital role in cell division, etc.
So microtubules,
Okay.
superhighways or train tracks, straight, rigid pathways, tiny motor proteins haul cargo, efficient distribution, also form backbone of cilia, tiny oars, and indispensable for cell division. Truly impressive, constantly active internal transport and structural system.
Absolutely, a dynamic, essential framework.
Wow, what an absolutely incredible, exhaustive journey into the cellular city. From the nucleus, our protected library and command center, to the RER's protein factories, the SER's chemical labs, then the Golgi's sophisticated post office, customizing, sorting, shipping, then lysosomes recycling plant, peroxisomes detox squad, of course the mighty mitochondria's power generators, and finally the amazing cytoskeleton providing structure, movement, transport. It's truly a bustling, incredibly organized metropolis, all happening in a space too small to see.
You've just gained a fundamental understanding of how billions of these tiny, intricate machines work together in perfect and dynamic coordination to create and maintain a human body. This deep dive into cell ultrastructure isn't just academic curiosity, it's the absolutely essential foundation for understanding every disease, treatment, biological process you'll encounter in medicine. Thinking about explaining it to a 10-year-old, finding those vivid anchors, forces you to truly grasp the core concepts. Move beyond memorization to genuine, applicable comprehension. That's a skill that will serve you incredibly well, not just in medicine, but in communicating any complex idea clearly.
So what does this all mean for you, the listener? As you study or just reflect on life, next time you look in the mirror, consider this. Every single cell in your body is performing these incredibly complex, coordinated tasks right now, in perfect harmony. If each cell is a city, and you're made of trillions, just how immense and intricate is the universe contained within you? And what might happen if just one tiny, perfect system starts to falter in a way we haven't discovered yet, or are only just beginning to unravel? The more we understand these microscopic worlds, the closer we get to unlocking the secrets of health and disease.
Keep asking why and how. The beauty and wonder of biology truly lie in these intricate details, understanding not just parts, but the magnificent symphony of how they interact. And remember, the elementary course materials we drew from. Fantastic place to reinforce these concepts, continue building your knowledge base, solidify these ideas. Until next time, keep exploring the incredible, ever-unfolding world within.